Hello guys, today we are going to talk about one of the most significant cells of the blood that is known as platelets. They are not complete cells, rather they are fragmented cells, but they are the players of blood coagulation. When bleeding occurs, after some time, a soft jelly-like mass is formed at the site of the injury and that is known as a blood clot. And this clot formation is mediated by these cells which are known as platelets. In mammals, in the circulating blood, these are known as platelets, but in the non-mammalian species, these are known as thrombocytes. You will be fascinated to know that platelets are fragmented cells. They do not have the nucleus, but they do have the mitochondria and canalicular system. But thrombocytes are complete cells. They are having the nucleus and distinct organization with a distinct elliptical shape. Whereas the platelets, they do not have any specific shape. They continuously change their shape even along the species. It also varies. The size also varies. Can you believe that 10 to the power 11 number of platelets are formed per day, but they are 1/10 to 100 of the RBC size. Platelets, they do not directly come to the blood circulation, rather they are stored in the spleen. With this basic introduction, let us begin. What are platelets, how the platelets originate, what are the platelet function, what are the platelet structure, and how these platelets play a key role in the process of blood coagulation. Before going into the detail, please like and subscribe my channel. So I will write some basic points what I said, and thereafter we want to go into the details. The number one point, these are incomplete cells. That means these are the cells which are fragmented from a big cell that is known as megakaryocyte. We'll see after some time. But these platelets are only found in the mammalian blood. This is a peculiar point. In the mammalian blood, these are rod-shaped, oval, sometimes spherical, even these are cylindrical in shape, means various shapes are there. In mammals, these are also said to be anucleated. That means the nucleus is said to be absent. But there is presence of cell organelles. And these cell organelles carry out various functions that we'll discuss under the structural part. Most important point what I wanted to convey you people is the thrombocyte word. This thrombocyte, these are found in the non-mammals and they are the same cell just like that of the platelets. But here these are elliptical in shape, if I can say elliptical in shape, and they are considered to be nucleated. And here these are showing complete organization. That means their cellular integrity is maintained. But what is the common point? The common point is that it may be platelets, it may be thrombocytes, both are associated with the process which is known as coagulation of blood or blood clotting. That means it may be thrombocyte, it may be platelet, it may be mammal, it may not be the mammals, but everywhere the function is blood clotting. That means the formation of a blood clot which prevents excessive bleeding from the site of injury. So this is the first flowchart which is showing a comparison between the platelets and the thrombocytes. Next come to know how the platelets originate. The platelets originate from the red bone marrow. It is said to be the site of synthesis of the platelets. Just like that of the RBCs and leukocytes, platelets originate from a multipotent cell which are known as the PHPC cell. Let's draw a diagram to explain how the platelets get originated and what are the dynamics of platelet origin. Pluripotent, hemopoietic stem cells. These are the cells PHSC. These are the cells which shows continuous process of division and self-renewal as well as differentiation into multi-prototype cells. These PHSC cells, they are said to be the stem cells which are first of all multipotent and they are present in the red bone marrow. So these multipotent cells are quite capable of differentiation as well as division. So they give rise to another category of cell which are known as the colony forming unit, spleen. The colony forming unit, spleen. And these cells are known as, I can say, CFUS. These are the cells which again shows formation of colonies and they are going to form a niche of cells or a specific category of cells. Those category of cells are known as colony forming megakaryocytes. Let's see, this is another group of cells which are known as colony forming unit megakaryocyte. Students, please remember, this megakaryocyte is the precursor of the platelets. This is the largest cell which undergoes fragmentation to produce 100 to 1000 of platelets. So this cell CFUM, this is the precursor cell which going to rise the next cell which is known as megakaryocyte. Megakaryocyte is the fundamental cell from which the platelets are going to originate. So this is one of the most important cells in the origin of the platelets and their formation. Try to understand a point. This total process is occurring in the red bone marrow under the influence of several growth factors, transcription factors, lineage factors, stem cell factors, and totally under the control of the genetic factors. No doubt about that. Interleukin 3 and a very specialized hormone which is known as thrombopoietin. This hormone is secreted from the hepatic cells and from the kidney cells or the renal cells which regulate this process of differentiation. Can you believe that these megakaryocytes, they are having the receptors for this hormone thrombopoietin, and when the number of megakaryocytes increase in the blood, then the thrombopoietin binds with them and provides a feedback to the red bone marrow to stop or to inhibit or to reduce the formation of these differentiation cells so that the normal count of the megakaryocytes will be maintained. That means indirectly I am saying it will maintain the normal count of the platelets. So this is a controlled, regulated process and strictly regulated by the circulating number of platelets as well as the expression of this particular hormone receptor that's known as thrombopoietin. So we should not forget this is the hormone thrombopoietin and one of the most specialized interleukins, that is interleukin 3. These are the these two molecules, they regulate, but this is that hormone thrombopoietin, the receptor that is regulating this process. Its origin is liver and from the kidney, which are regulating the process of thrombopoiesis. This hormone maintains the normal circulating number of the platelets. Then these megakaryocytes undergo fragmentation, and this fragmentation produces the ultimate circulating cells or ultimate cells which are known as the platelets. So this is the process of origin. During this process, platelets, each megakaryocyte may give rise to 100 to 1000 number of the platelets. And during this process, what happens? Now these megakaryocytes, they show pseudopod-like process in the cytoplasm, and these processes, they undergo pinching up or budding off. They undergo the process of boarding off, and then they are released in the form of the platelets. So guys, you have to remember that during this process of pinching up of the boarding up under the influence of this thrombopoietin and interleukin 3 and several growth factors, these platelets are formed. And nevertheless, these platelets are stored in an organ, that is spleen. It ranges between 30 to 45%. That's why in the normal circulation, you will not get the adequate number of platelets. Please remember, the normal count of the platelet is ranging between 1.5 to 2.5 million per cubic ml of blood. But during the time of injury, these platelets get come into the blood in a rigorous manner, and that release is mediated from the spleen. That's why spleen is known as the reserve of the platelets, and it also regulates the circulating number of platelets. So this is in general how the platelets are being formed in the red bone marrow. We should not forget the role of the thrombopoietin. So this is always a question. Name the hormone which is responsible for thrombopoiesis. Answer is thrombopoietin. Number 2, who serves as the reserve of the platelets? Answer is spleen. The third point, how many days it takes for this thrombopoiesis process? So it takes around 3 to 6 days. And this is another point. DAS is said to be the major site of thrombopoiesis. Answer is RBM, red bone marrow. How many number of platelets are formed from one megakaryocyte? It may be ranges between 100 to 1000. Name the fragmented cell present in the human blood or animal blood. The answer is the platelets. So like this, several questions will come. And what is the normal count? It ranges between 1.5 to 2.5 million or 3 million per cubic ml of blood. That's also important to remember. So after this, let's further dig into the details, that is the ultrastructure of platelets. Why I am telling all these stories to you? Because we are going to discuss about the clotting mechanism, clotting cascades, platelet activation mechanisms, formation of primary blood clots, which are directly associated with the platelet function. So let us begin to understand the structure of the platelet, and then the third part we will come to know about the functions of the platelet. Please do not forget that platelet is only having one function, that is clotting of blood. Rather, I can write 10 functions which will define how platelets are so important to us. Let's see an ultrastructure of the platelet. We do not give emphasis on the platelet structure, but we should know how a platelet is formed. Thereafter, we can understand the functioning of the platelet in detail, because platelet shows so many number of receptor molecules. Can you believe that platelets are said to be the reserve of multimeric signaling molecules? I can say it's a signaling reserve. But do not forget that it is not having a complete organization. Rather, multiple parts organelles are being missing. Platelet, just like that of the normal cells, also utilize the energy. Mitochondria is fully functional. Calcium is the regulatory factor for the platelet function. It also regulates the degranulation process of the platelets. The cell membrane of the platelet shows multiple number of receptors which act as the site of binding to the endothelium, the site of binding for the collagen, the site of binding for the von Willebrand's factor. That means platelet is really said to be a cell of choice to study signaling. Number 2 point is that platelet has evolved some of the structural components which are never seen in case of erythrocytes and leukocytes. That makes platelets very complex. Next, the third point is that platelet stores glycogen. A lot of glycogen granules are stored and they act as the source of energy to the platelets. Platelets do so lysosomes which are storing the acid hydrolysis, acid phosphatases and other type of digestive enzymes which are required for the platelet function. Never forget that platelets do also have actin and myosin like contractile filaments just below their cell membrane. Their cell membrane is made up of phospholipids and glycoproteins, and it commonly said to be a glycocalyx. It also stores the prostacyclins, it also stores the echinocyanins, and it synthesizes arachidonic acid and the prostaglandins. That means platelet is truly a cell even though it is not having nucleus. But do you know that the platelet reserve is only for 7 to 10 days? That is the circulating half-life period of the platelets, and after that the platelets are detected by the macrophages of the blood and get destroyed. So this makes platelets a very interesting cell, and we should not forget that platelets are the major mediators of physiological functions of the blood. Let's draw a diagram where we'll show how the platelet is being organized. We'll touch every point and then we'll go to the last part, that is the platelet function. I am drawing a platelet easy to show the different parts. Here I am drawing a spherical structure, but remember, it may be spherical, it may be elliptical, it may be oval, it may be irregular, it may be bordered, it may be studded. So let's see how it is. So it shows a thick layer just below this. This is the cytoskeletal element of the platelet, which is present just below the platelet cell membrane. You are seeing some of the granules which are very, very dense. These are dense granules. Here you can see in the pink chalk what I have drawn. These are the dense granules. And we'll see what do you mean by these dense granules, what do you mean by alpha granules. Presence of some of the bigger means a big size granules are there like this. These are big size granules are present, and for your information, these are known as the alpha granules, one of the most important type of granules which are present inside the platelet. If you can ask me the number, this is 220 to 200 per cell. So you can imagine how complex these granules are. But if you go to the dense granules, these are hardly 2 to 10 per cell. That means their number is less. And these are the granules which are the storehouse of these mediators which make the platelet very important. We will compare what are dense granules, what are alpha granules. Remember, these alpha granules, they contain the high molecular weight compounds. I will draw another comparative chart where I will show what are those high molecular weight compounds. They mostly contain, the dense one, the low molecular weight compounds. Here, if you see the low molecular weight compounds, what they include? They include the nucleotides, the adenosine nucleotides. Adenosine nucleotides. They contain the serotonin, they contain the calcium, and you can see these are the metabolic regulators. I can say these are the metabolic regulators. That means the ATP functioning mechanisms are also regulated by this. The serotonin is the vasoconstrictor that's also present in the dense granules. But when I'm telling about the high molecular weight compounds, then we should remember that it is containing the factor like fibrinogen, which is the clotting factor number 1, and it is a major mediator of blood clotting. Then the accelerine, the factor that is 5, this is factor 1. Next, one of the most important bond with brands factor. This Bond-Will-Branch factor is the this is Bond-Willebrand factor, Willebrand factor. This factor is associated with adherence of the platelet. That means the deficiency of this Bond-Willebrand factor leads to poor attachment of the platelets, and then the platelets are failed to form the platelet plug. Even in the deficiency of this, the platelets are failed to show the process of activation, and therefore the platelets are failed to aggregate. Even the platelets are not attracted to the site of injury. Even they are also not attached to the exposed collagen surface. So that makes these granules, alpha granules, and their constituents important as the signaling molecules. These are the signaling molecules, but here these are regulating the metabolic function. Now coming to the third part, you are seeing the platelets which are having the powerhouse of the cell, and these are known as the mitochondria. And the ATP generation mechanism is completely regulated by these mitochondria. They are also serve as the source of reserve of calcium which are required for mitochondrial function. Then you are seeing circular structures which are important as suicidal bag of sac. These are lysosomes, and these lysosomes are the store house of acid phosphatase, the rate limiting enzyme of the platelet function. Acid phosphatases, alkaline phosphatases, and other digestive enzymes like hydrolysis. These are also present here inside the lysosomes. This is another very important point. Then you are seeing some of the canalicular structure. These canalicular structure, I can write, these are known as the tubular structure of the platelets, the tubules, the tubular structure of the platelet. And can you believe that these tubular structure of the platelets, they also store the calcium. That means these are calcium storing unit. They are the regulators of release of the calcium during the process of this platelet activation. Then you are seeing some of the structure which are appearing like canalicules. These are open canalicules which are spheres inside the cytoplasm, and they are open to outside. These are known as open canalicular system. This open canalicular system are those which carries this alpha granules in them, and it shows this removal of the alpha granules as well as their release. That means these are the major regulators of degranulation of the platelets and the release of these granules. Thereafter, I am seeing the glycogen droplets. There are so many glycogen granules which are present inside the platelets. These are the glycogen granules which are the source of the energy to the platelets. So glycogen granules are also present inside the platelet. Now you are seeing the platelet membrane. This is made up of phospholipids. This is important. This is made up of glycoproteins. This is made up of glycolipids, and they are said to be the membrane regulators, or I can use another word, that is glycocalyx, that makes the surface of the platelet negatively charged, so that the normal circulating blood vessel with an intact endothelium, the platelets do not attach and do not come in contact with the endothelial cells. Thereby, they do not adhere to each other to initiate the process of auto-clotting. And this makes the platelet membrane very dynamic. Then you are seeing the actin myosin like filaments which are interspersed just below the surface of the cell membrane, and these are the contractile elements, and they are the actin and myosin. This is actin protein and myosin protein, which makes the structure very, very complicated, very, very contractile. Remember, platelets do not move. They don't move out of the blood vessel to reach the tissue spaces. No, it is not that. But the degranulation process, the calcium dependent degranulation process, requires active contraction of the platelets. And this active contraction of the platelets help the process of degranulation. Calcium and ATP from the mitochondrial source and from this canalicular system, that is tubular structures which are present, that is these are dispersive canalicular structures. They are the regulators of the release of the calcium, by virtue of which these vesicular structures, these granular structures, they contract, and by virtue of this they release their constituents to outside. Besides this, the membrane is showing the receptors, those receptors which are going to bind with the endothelial surface as well as various activators of the platelet. That is just like the factor number 2b, the factor number 3a. So these are some of the factors which are coupled to G protein. That means they are GPCR receptors. When the ligands are attached, This GPCR signaling gets activated, by virtue of which the calcium and the mitochondrial pathways get activated, and there is release of the platelet constituents just like thromboxane A2, and that thromboxane A2 and serotonin is going to show vasospasm or vasocontraction, but thromboxane A2 and ADP, the molecules like ADP, because I have written here ADP, they are going to activate the clotting cascade. Once one platelet is being attached to the site of damage to the endothelium, then it will activate further the platelets, and that process is known as platelet activation. This activation causes further secretion of a factor that is known as platelet activating factor, that is PAF. This platelet activation factor or platelet aggregation factor attracts the further platelets to come to the site and click to each other, so that they will form a plug or a clog, which serves as the site for the deposition of the fibrin threads, and a stabilized clot is formed. To know the details about the platelet activation, platelet adherence and platelet pathways, you have to go to the next lecture, that's known as the blood coagulation pathway or the hemostasis pathway. So with this structural part comes to the end, and this structure shows platelet, even though is not having the nucleus, but it is one of the most complicated cells which is present in the blood, and the average lifespan of the platelet, it serves as 8 to 10 days, and after 8 to 10 days the platelets are destroyed and they are trapped by the macrophages, which are said to be the cells which are associated with the destruction of the platelets. So with this, we come to the conclusion of the granules, the ultrastructure of the platelets. Now, I'll write down the final flowchart, that is platelet function. And after that, this lecture will come into a conclusion. And the next lecture is about the mechanism of hemostasis, the 4 definitive steps of hemostasis, and then we'll go to the intrinsic and extrinsic mechanism, where we will learn how the prothrombin activator is formed, which converts the prothrombin to thrombin, fibrinogen to fibrin, and ultimately the fibrin is stabilized to form the blood clot. So let's draw another picture, that is functions. So this will come to the conclusion. The platelets, the physiological function, the number one, it is the storehouse of clotting factors. This is one point. The second point, blood clotting, or it mediates the coagulation of the blood. The third point, it keeps the factor number 8, anti-hemophilic factor, intact. That means it doesn't allow its degradation, because you know that this binds with the von Willebrand's factor and it gets stabilized, and platelet serves as the site. Then it secretes platelet growth factors. These platelet growth factors, they are associated with mitogenic functions. That means division of the cells. Mitogenic functions. They also help in the process of healing of wound. That is another important function. Third function, they are associated with the capillary growth. They are also associated with the endothelial growth. That means these are very, very important function, endothelial growth. Next, I am coming to the next function, that is clot retraction. This is a process in which the clot is retracted so that the serum comes out of the clot and clot gets hard, stick, and becomes stable. And that clot retraction is mediated by the contractile proteins which are present in the platelets, platelet contractile proteins. And these platelet contractile proteins, like you have heard this word thrombocytosinin. This protein, it causes the contraction of the clot and serum comes out of that. These platelets, they are also containing the activators of clotting cascade like the thromboxane A2. The thromboxane A2, this is a very important function. Then they also contain the platelet factor C. This is another important factor. They also contain the von Willebrand's factor, which is required for platelet adhesion. That means these are some of the very, very important functions of the platelet. Then the next function I'm writing, that is platelet activation, platelet adherence, and third, the platelet clot formation. All these are the functions of the platelet granules which are being present. Then the first process of blood clotting means when the blood vessel is damaged, that is vasospasm. This vasospasm is mediated by the serotonin and that is also regulated by the platelet. Platelet also regulate the endothelial function. This is another important function, endothelial function. It also maintains endothelial integrity and it behaves the endothelial to maintain the smoothness throughout the course of the blood flow. So this makes the platelets so important for us because platelets contain prothrombin activators also, and these prothrombin activators along with these icosanoids, that means the arachidonic acid, the prostaglandins, they are also released by the platelets. That means the platelets also release the prostaglandins. This is another important point, prostaglandins. The platelet also contain the glycoprotein factor number 2 and factor number 3A. Please remember, these are the sites of the GPCR, the G protein coupled receptor signaling, and this leads to the platelet activation and release of the thromboxane A2. It also causes release of ADP, that which is leads to the platelet adhesion. So we should not forget that it contains the collagen receptors. Means when the blood vessel is damaged, there is exposure of the endothelium, collagen is exposed. To this the platelets go and attach, and that makes the platelets a cell of choice. So thank you so much, guys. Extensively we have discussed the platelet. Next class is the fundamental phases of the blood clotting. So stay tuned for my next lecture where I am coming with some of the very, very fascinating questions of physiology related to the blood vasculature and how the process of clotting occurs. So let's tune for my next lecture. Thank you so much and have a clear understanding of the concepts from PhysiologyEduHub by Dr. Dilip Swain. So please like and subscribe my channel. Thank you so much and have a wonderful time.